Cool. Thanks very much to the Rising Stars Committee for giving me the opportunity to talk today. And I'm coming to you from Gadigal land here at the Victor Chang. So yeah, as Angelo introduced, I'm going to be talking to you about some multi-omics work that we're doing to try and look at the function of perhaps the most important gene in vascular disease. And this gene is factor 1. Now, factor 1 was first identified by Professor Kovacic along with international collaborators back in 2016 as being linked to fibromuscular dysplasia. It was found that patients with fibromuscular dysplasia had increased expression of factor 1. Now, the expression of factor 1 is governed by a single nucleotide polymorphism within the gene that can encode for an A or a G at this position. And this is an expression quantitative locus. So when you have the AA allele, you have higher expression of factor 1. And when you have the GG, you have decreased expression of factor 1. And subsequently, it's been found that factor 1 increased expression is not only linked to FMD, but it's also been shown to correlate with migraines and also the development of spontaneous coronary artery dissection. Now, what's unique for factor 1 is, while increased expression of factor 1 is related to one subset of diseases, decreased expression of factor 1 is related to a whole different genre of disease, where we see that decreased expression is linked to the development of coronary artery disease, as well as myocardial infarction and arterial stiffness. But despite the clinical importance of factor 1, the way that it signals within the vasculature is poorly understood. And what we do know is that the factor 1 gene encodes for the phosphatase and actin regulator 1 protein. And on the surface, it kind of does what it says on the tin. It binds to protein phosphatase 1 and targets it to substrates to regulate phosphosignaling. But this is a reciprocal relationship with binding to G-actin. And the binding to G-actin and PP1 is known to regulate the actin cytoskeleton, stress fiber formation, which together regulate cell motility. Now, factor 1 is expressed in most tissues within the body, predominantly in the brain, but also in major vascular tissues and blood and immune cells. And to complicate our lives, there are a lot of isoforms of factor 1. Within our lab, we've discovered at least 21 different transcripts, 18 of which encode for a protein product. But for the purposes of today, I'll be concentrating on this factor 201, which most people regard as full length or the kind of common factor 1 transcript. So when we create factor 1 stable cell lines, either with knockdown or overexpression of the gene, we see that there's this distinct change in cell morphology. And this is unsurprising given its known binding partners. We see this change in the actin cytoskeleton, as well as changes in lamellipodia formation. But what I'm really interested in is what else is going on in these cells and how that leads to the development of vascular disease. And to look at this, I'm taking this multi-omics approach. Now, we all know that you have your genes, which then go to your transcripts, to your proteins, and then resulting from the protein enzymes, we get metabolites formed, and also, importantly, lipids that make up your cell membranes, as well as other important signaling molecules. And we can use different omics techniques to look at every single step of this pathway. And by doing that, we can understand the flow of information that's governed from the factor 1 gene. Now, we can take these omics individually, and the kind of popularity and the access to each of these techniques has been growing over the last 15 years. But what's really interesting to us is this kind of multi-omics approach. And this provides this really unbiased approach to give us these multi-level insights. And what we hope to gain from this is a very broad, unbiased understanding of how factor 1 signaling is impacting these cells and your vasculature, and how that's leading to the development and progression of disease. Sorry, so just in the interest of time, today I'm going to talk to you about a project of mine that's using genomics, transcriptomics that we've recently published, and then bolting onto this some proteomics work and a little touch on the others right at the end. So in parallel to our interest in factor 1, we're also interested in vascular disease in general. And with the New York part of Professor Kovačić's lab, we were performing these integrative studies to try and identify and prioritize genes that are causal for coronary artery disease. So with this study, we took GWAS data from the UK BioBank and also cardiogram and integrated it with these StarNet transcriptomics data sets. So this was data from 660 patients with CAD as long as healthy controls, and it looked at the expression of genes in 9 cardiovascular or CAD relevant tissues, which includes the aorta, the mammary artery, along with non-CAD tissues. And the expression of each of these genes is known, and then we can integrate that with our GWAS data and come up with these tissue-specific causal candidate genes. And performing this analysis, we identified 162 genes which are causal for CAD, and unsurprisingly, factor 1 popped up. And what was interesting was we showed that that factor 1 was causal, so it had its effects in the aorta and also the mammary artery, showing that it's having its effects within the vessel wall. Which really informs our studies going down the line, knowing that we should be looking within the vasculature at what factor 1's doing, and this is perhaps not a systemic effect. And when we ranked these causal genes, effectively factor 1 came out as the top ranked gene, so the most significant gene in for causing CAD. And as part of this study, we performed these co-expression networks where we looked at our key genes and we looked at what was in their second and third order of influence. And when we zoom in for factor 1, you can see that there's only one gene in its second order network, and this is ACAP1. And we were able to validate the relationship between factor 1 and ACAP1 in a second transcriptomics dataset, our GTEx dataset, whereby the expression of ACAP1 and factor 1 correlates in the aorta and the coronary artery, as well as other vascular tissues, but we didn't see this relationship in any non-vascular tissues, which suggests that ACAP1 and factor 1's relationship is specific to the vasculature. So in parallel to this, I was doing some proteomics work in general, just to see, if I change factor 1 expression in my HT1080 stable cell lines, either by overexpressing it or knocking it down, what changes on a global proteomics level? And in a nice validation of what we see in the human arteries, you can see that when we increase factor 1, we get an increase in ACAP1 expression. And I've confirmed this now an mRNA and a protein level within these cells. So what is ACAP1 and why would this be important potentially in vascular disease? So ACAP1 is a kinase anchoring protein 1, and its main role, or its most studied role, is in the targeting of protein kinase A to the mitochondrial membrane, where it's known to regulate ATP production, but this binding of this complex or formation of the complex also maintains mitochondrial integrity and inhibits mitochondrial or cellular apoptosis. But ACAP1 has other functions. It's also known to bind to multiple different mRNAs, which influences their expression inside the mitochondria. And this includes SOD2, which encodes for superoxide dismutase, the mitochondrial version of this. And this is involved in the regulation or removal of mitochondrial reactive oxygen species. But it also is known to regulate mRNAs that control ATP production and also lipid synthesis. And importantly for us, ACAP1 has a binding site for protein phosphatase 1. Now, I told you earlier that protein phosphatase 1 is targeted to its substrates by factor 1. So we can infer from this that factor 1 is likely part of this ACAP complex. So there's nothing known about what factor 1 might be doing within these circumstances, but given the relationship between the expression of these proteins, we could perhaps suggest that factor 1 might be stabilizing these ACAP complexes. And what would the impact of that be? Well, when we have these stable complexes where factor 1 is expressed or even has increased expression, we have the ability to maintain this mitochondrial integrity, as well as, stabilize these superoxide dismutase, increasing its production within the mitochondria, thus reducing mitochondrial ROS. But also we know that this complex is involved in the regulation of ATP production. Now, if we disrupt this complex by removing factor 1, and bear in mind that reduced factor 1 is causal for CAD, we see that potentially we get disruption of these complexes, which leads to the loss of regulation of PP1 and protein kinase A, a decrease in SOD2, which might feed into increased mitochondrial ROS, as well as potentially dysregulated ATP production, which can also impact ROS. And overall, have we changed the integrity of our mitochondria? And together, this really can feed into oxidative stress that is known to contribute to CAD progression. So in terms of evidence from this, so firstly, within these cells, when we overexpress factor 1, we get a stabilization or increased expression of SOD2. And when we knock down factor 1 compared to their controls, there's decreased expression, indicating that this might be the case. Now, using the Agilent seahorse to measure cell respiration and ATP production, can see that changing the expression of factor 1 significantly alters the ability of mitochondria to produce ATP. So we get a decrease in production of ATP in the overexpressors and an increase production of ATP in the knockdowns. I think what's interesting is actually, you can see the variation here. So there's kind of, where it's very tight in the other conditions, potentially disrupted or, dysregulated ATP production in this case. And something that some new data that I've just received is I was doing targeted lipidomics in collaboration with the Baker Institute, Peter Minkel down there. And what I noticed was that there's actually a change in the lipid composition within the mitochondria, whereby there's an increase in the key component of the outer mitochondrial membrane and the overexpressors. And this lipid is actually converted to the lipid that's a key inner mitochondrial membrane lipid with cardiolipans, and there's a decrease in this in the overexpressors. And this suggests there might be some kind of change in the lipid metabolism, these interconversions, the way that these cells are requiring lipids to build new membranes. So this is something that's really new data for me, but very interesting in terms of the mitochondrial story of what on earth factor 1 is doing to the mitochondria and how this is regulated by ACAP1. So overall, the next steps in this project are really to decipher the molecular relationship between these two proteins. I know from some co-IP work that we're doing that we don't see any evidence that factor 1 comes down in a complex with ACAP1 so far. So this is something that we're investigating more closely, potentially looking at some cross-linking experiments to see if we can capture this complex and see what's involved in it. Also key to this is actually looking at mitochondrial ROS production to see whether indeed, the change of SOD2 expression is linked to increased or decreased ROS production within the mitochondria. And then because of this observation in the lipid composition within the mitochondria and these cell lines, we want to see if this has the ability to alter the morphology of the mitochondria. kind of get a better understanding of what exactly these interactions are doing. Next, then we want to move this into our factor 1 knockout mouse model to validate that we see this same relationship in vivo, especially within the vasculature. And then using a high-fat model, we can see if this impacts the development of CAD within these mice. And finally, the kind of wider part of my project is, I'm currently generating transcriptomics, proteomics, metabolomics, and lipidomics data sets. And as well as the ACAP project, we're constantly integrating different parts of these omics to try and understand the wider role of factor 1 within the vasculature, and how this is contributing to disease progression. And with that, I'd just like to thank the Kovacic lab within Sydney, especially Jason, his lab in Mount Sinai, the Victor Chang Innovation Center, where we're performing lots of our mass spec, as well as our industry partners, Agilent Technologies, for all their help and support. Thank you.
Thank you very much, Kathyryn, for the very interesting presentation. We'll open up to questions, but I want to start because I have a question pertaining to your use of the word causal, and it might be just because I'm ignorant on the subject. But what is to say that factor 1 is causal in, say, the development of CAD, rather than it might be associated with another thing that impacts factor 1, and then through that it can influence CAD development. So basically what I'm saying is, how do you know that factor 1 is actually causing CAD and is not being contributed to by some other factor? And I don't use that word as in like, and I use FA.
Yeah. Absolutely. Yeah, no, of course, that's interesting for us as well. And really this comes down to the use of our of StarNet and the different tissues that we used in StarNet. So as well as having the aorta, which forms atherosclerotic plaques, we also have the mammary artery, which doesn't form atherosclerotic plaques. So we can look at the relationship between these between the gene expressions in the different tissues. And essentially what the mammary artery provides us is a vessel that essentially we can class as pre-atherosclerotic. So it hasn't actually, it's in the process of developing this disease, but hasn't actually got there yet. So so the expression of the tissues and expression of the genes and the changes in the genes in that mammary artery within the field are considered to be linked to causal genes.
Yeah, I think again it might be just because I'm not up to date with the genetic literature, but yeah, just I was wondering how much can factor 1 be affected by lifestyle behaviors, or is it that you are born with a certain amount and because of that then you have a certain you have a higher or lower risk for CAD?
Yeah. So that's where the SNP comes in. So everyone has one version of the SNP. So you will either be AA, AG, or GG, and it's this presence of the AA or the GG that governs the expression of factor 1. Whether other factors contribute to the expression of factor 1 and impact, such as lifestyle factors, that isn't something that we have extensive data on at the moment. But I think what kind of is very clear, is the multitude of genomic studies. It's not just us that's perform these large-scale genomic studies that have linked factor 1 to vascular diseases, but there's now kind of 12 studies out there all looking at different vascular diseases that have all come to the same conclusion, that this gene and different versions of this gene are very important in the development of a plethora of different vascular diseases.
Oh, yeah, super interesting, because I know that reactive oxygen species or inflammation can also be contributed by so many different things. So
yeah, absolutely. Yeah. I mean, this is with that kind of part of it, it's very early on, and we kind of are
untangling everything. I get you.
Yeah, absolutely. It's a very complicated story, and we're trying to just do it one strand by kind of one at a time, bit by bit. But yeah.
Thanks, Kathyryn. So look, we have 2 questions from the chat. Peter asks, is your mouse model a straight knockout? Oh, sorry, which cells express factor 1, and is your mouse model a straight knockout?
So in terms of cell types, most cell types express it to a certain degree. We in the mouse, currently we're in the process of kind of characterizing our mouse. And what we're seeing, at least from a mouse side of things, is you get lots of expression in the brain. There's some expression in the vasculature and these inflammatory tissues. You also get quite a lot of expression in the pancreas. We don't see much kind of in other tissues like liver. In terms of how we made the mouse, it's actually a long story behind it, because I showed you there are all those different isoforms of factor 1. And essentially, if you just knock out 1 or 2 isoforms, so if you kind of create a conventional knockout mouse, you basically can't get rid of factor 1. So what we've had to do is actually take out the entire gene. So it has the first exon, and then it has none of the rest of the gene. But we have the constitutive knockout, but then we also have a floxed version that we are generating smooth muscle cell and endothelial cell specific knockouts. But yeah, that's kind of exciting work that's in the pipeline. I'm sure you'll be hearing Kovacic Lab talk about soon.
Thanks, Kathyryn. And really last question. So this is from Monique Bax. Given the involvement of SOD family in motor neuron disease, are you planning on looking at the impact of changing the regulation of factor 1 and ACAP1 on motor neuron health?
So yeah, the role of factor 1 in the brain, as I've mentioned, there's a lot of factor 1 in the brain, and kind of it's really interesting where it's expressed in the brain. So it is definitely something that we are constantly thinking about. In terms of our expertise, we definitely aren't the lab to look at factor 1 in the brain, but that's something that I think down the line we'll be getting collaborators in and involved in. And if anyone has suggestions of nice neuro labs that might want to work with us, that'd be great.
I know a couple.
Yeah.
I'll have a chat to you after, Kathyryn.
Yeah, for sure. Thanks, Monique.
Thank you, Angelo. That's great. Good afternoon, everyone, and thank you very much for the opportunity to present to you today. Today, I'm going to talk to you about advances in congenital heart disease genomics, and it's a very broad, overarching title, but I thought it'd be nice to give you a bit of an overview of our research program, which focuses on congenital heart disease and specifically on genomics, but also take you on a bit of a journey through the last decade where we've really seen massive changes that have actually translated into the clinical sphere. So I thought, hang on, there we go. There we go. So I thought I'd start off just talking a bit about congenital heart disease for those of you who aren't familiar with it, and talking about some of those changes in the last sort of decade. I then thought I'd talk about a study that we've very recently published. So it's hot off the press, was published about 2-3 weeks ago. And then finally end off talking about the implications for patients and really talk about that translational impact from our work. So what do we know about congenital heart disease? Well, it's a structural abnormality of the heart and great vessels that is present at birth, and it occurs in about 1% of the population, so 1% of live births, and it really is the most common birth defect that we see. In Australia, about 3,000 infants are born with congenital heart disease each year, and a third of these will require surgery within the first year of life, with some of them requiring multiple surgeries and intensive care throughout their lives. So it places a significant burden on the healthcare system, but also on the family and very much on the individual throughout their lives in terms of the social, financial, but also psychological perspectives. And the key questions that parents ask upon a diagnosis, which is usually either prenatal or very early on when the baby is born, is why and how did this happen? And how do I stop it from happening again? And this is really, I guess these questions are really what drives all the research that we do. So what do we know about the causes of congenital heart disease? Well, prior to sort of the genomics revolution, we used to say that we know the cause of about 20 to 30% of cases we see, but for the vast majority, about 70 to 80%, highlighted in grey, the cause was unknown. And it was assumed to be multifactorial in origin, so implicating multiple genetic factors and environmental factors in disease causation. However, in the last decade or so, these significant advances in genomic technologies have begun solving this unknown portion so that we're now sitting roughly at half known and half unknown. The contribution of copy number variants, aneuploidy, and environmental factors have remained largely the same. And what's really changed is this significant contribution from transmitted and de novo variants that has come through these genomic studies. So this is actually a bit of an older slide now, but it, I think, nicely shows a timeline of sort of the discoveries in the field. And you can clearly see, I'm not sure if you can actually see my mouse if I'm pointing on this slide, but basically you can clearly see from sort of 1990s to 2000 genetics coming into play with the first gene being discovered. And then in 2010, you can really see that explosion of activity with genomic technologies coming into play. And this has really resulted in a number of landmark publications and studies where large sample sizes were combined with sophisticated statistical approaches. And this really has enabled very important and novel discoveries in the field. And I'm not going to go through, I know many of you from different areas, but I thought I'd just quickly highlight some of these key discoveries in the last sort of decade. So what these studies have shown us is that there are likely to be about 400 genes that cause or contribute to congenital heart disease. And to give you an indication, I will touch on this a bit more in a later slide, but at present we know about 139 of these genes. So there's still a long way to go. And what the sort of research is pointing at is that they're very likely, the contributing genes are very likely to be chromatin modifying and cilia genes. What's also come out of this work is that syndromic cases of congenital heart disease, and when I talk about syndromic, I mean patients who have a heart defect as well as an extra cardiac anomaly such as a renal or kidney defect. Very often those variants were de novo variants and chromatin modifying genes. And for those patients who had isolated congenital heart disease, so they had no other health conditions, they just had a heart condition, they found that in many of those cases, they were inherited rare damaging variants. So they actually came from a parent very often who was unaffected. The contribution of common variants have also been highlighted, and quite importantly, there's been evidence to suggest a genetic link between heart and brain development, which is really important because we've always known that patients with congenital heart disease are at higher risk of a neurodevelopmental disability. So as I mentioned, we now know of about 139 genes that are linked to human congenital heart disease. And basically these genes have been reproducibly shown to cause human congenital heart disease. And there's a very handy website that we've developed with our team at Victor Chang called CHD Gene. And basically this lists all the genes that are currently known to be associated with congenital heart disease. It's also very recently been published, so it's now citable as well. But it's a great resource. So if you have a heart condition and you have a pathogenic variant in one of these genes, it's very likely to be the cause of your heart condition. So moving on to our recently published study, and as I said, it's very much hot off the press, published in the last couple of weeks, and this work was basically the foundation of my Heart Foundation postdoctoral fellowship. So a big shout out to the Heart Foundation for supporting this work. But it really came from being in the field for 10 plus years and noticing that all these children are obviously growing up and they're going to have children of their own. And this growing patient group, as it approaches reproductive age, the causal genes that are contributing to the heart condition will be passed on to subsequent generations. And you can see that very nicely here. This is a family of ours in the corner of the slide where dad had a significant heart condition repaired as a baby, and he's gone on to have a little boy who has an even more significant heart condition and will need lifelong care. And I guess I saw a lot of this happening and thought we need to start doing something to look at the next generation. So what's going to happen to these kids when they grow up and they want to have their own children? And in line with this, there's been a lot of work that really shows this exponential increase in congenital heart disease among adult population, and that now this significantly outnumbers the pediatric population. So what I wanted to do is try and understand what's causing it. Are there any differences between the severity of the defect so that we could potentially modify the expression of the heart defect so that, or lessen the effect, or ideally so that these children don't end up having kids with heart defects themselves. So the overarching aim of this work was to investigate the genetic composition underlying congenital heart disease severity using whole genome sequencing. And to do this, we used whole genome sequencing, and we had two groups of participants. We classified them as either having complex or severe congenital heart disease requiring neonatal surgery. And then the other group were those who required surgery after the neonatal period, so had simpler forms of disease, if you want to call it that, a very loose term. We conducted a principal component analysis to remove outliers, just to make sure that we could actually, there was some validity in our comparisons, and we had to remove 28 of our participants to ensure we could adequately compare those two groups. As mentioned, whole genome sequencing was performed, and the initial bioinformatics analysis, we focused on those high confidence CHD genes that I mentioned earlier. At that stage, there were only 107 of them, not 139. So this analysis focused on those 107 genes. After that analysis, we then extended the analysis to the exome. So we opened it up, the analysis looking at all the genes across the exome. We performed Poisson log linear regression to compare the genetic variance between the two patient groups for both the high confidence analysis and the exome analysis. And then we used Fisher's exact test to determine the variant burden. So when we look at the study participants, and I want to highlight that this work is very much a preliminary study. The sample numbers are very small, as you can see, and we were limited because of the whole genome sequencing and obviously the cost thereof at the time. But we did see, we had no significant differences between the groups when it came to clinical and demographic factors, as you can see here. However, there was an obvious difference in terms of their CHD type, with those having more severe disease, having more patients with functional single ventricle, which is probably the most significant heart defect you can have. And we obviously purposely selected for that. And conversely, those patients in the other group had significantly more with simple heart defects, such as a hole in the heart and septal defects. So when we performed our analysis, looking at, or really limiting it to the high confidence congenital heart disease genes, which, just to remind you, are those 107 genes that cause congenital heart disease at the time that we were aware of, you'll notice that there was a significant increase in common variants. Now, common variants are those to have a minor allele frequency greater than 0.05. Found a significant increase in common variants among patients with severe forms of disease as compared to those who had simpler forms of disease. And conversely to that, we found that patients with more severe forms of disease had significantly less low frequency and rare variants compared to those children who had simpler forms of disease. So as mentioned, we then thought, let's extend, this is amongst the heart genes. So we thought, I wonder what's going to happen if we extend this out to the exome and look across genes more broadly. And when we performed that analysis, we found that the same trend held for rare and low frequency variants, in that the more severe forms of disease had significantly less rare and low frequency variants than those with simpler forms of disease. And when we looked at the common variants, however, there were no significant differences. So what does all of this mean? And I guess we were interested to know, are there any specific genes or variants that, or pathways that could be contributing to these differences? Are there potential modifying pathways that we could look at? So we performed a Fisher's exact test to compare the variant carriers between the two groups. And when we did that across the high confidence genes, so these are the genes that we know cause congenital heart disease, several genes actually reached nominal significance. However, when we corrected for multiple comparisons, only those common variants in CAT6A, which is a chromatin modifying gene, actually remained significant. And this was quite interesting to note because CAT6A is only recently discovered. So there's probably it probably has more of an involvement than we realize. When we looked across the exome, so we focused on rare and low frequency variants because they had the significant differences between the groups. We used Fisher's exact test again, and we inputted those significant genes into ENRICH-R. So this is a program that looks for associations between gene sets and pathways. And what came out, and it might be a bit too small for you to see on the slide, but basically the significantly impacted pathways in the KEG21 pathway system was autoimmune thyroid disease and type 1 diabetes mellitus. And in the DB gap was socioeconomic, smoking, and lupus. And what really stood out for us is that all of these factors are known risk factors for congenital heart disease development. So a particularly interesting finding to come out of this work. So in conclusion, and again, I'll highlight that this is very preliminary. It seems the genetic architecture of complex critical congenital heart disease, so severe forms of disease, is distinct from other types of congenital heart disease in that there is an increase in common variation that is specific to known or established congenital heart disease genes. In comparison, milder forms of disease have an increase in rare and low frequency variants. And I've tried to highlight that here in the diagram just to, I know it's quite a complex topic to grasp, but essentially as your disease increases in severity, you see that you have a genetic makeup which looks a bit more like this, a lot more common variants contributing to that final disease presentation. And then if you have milder forms of disease, you sort of have more, you have rare and low frequency variants of greater effect that seem to be contributing to that disease. What also came out of it, as I mentioned earlier, was this associations with regulatory genes such as CAT6A, but also environmental stresses and the role that they may play in the final presentation of disease. So the next step really is to replicate the findings in a larger group, and we're actually, as part of a separate study that we're doing with Sally Dunwoody at the Victor Chang, is we'll be sequencing, exome sequencing 950 of our patients. So I'll be using that as a replication cohort to see if we get any more information and in regards to what we've seen in this preliminary work. So that's the part I was going to talk about our current study that's just been published. I thought I would now move on to what this means for patients and really what is the effect that we've seen after 10 years of doing work like this. So when I first started and prior to the genomics revolution, we really had no pathways for testing for patients with congenital heart disease. They were just assumed to be unknown cause and you're just going to have to learn to live with it, and that's just how it is. Whereas now, thanks to all these significant advances that we've made, we have established testing pathways for these patients. And we, as you can see here, this is just one of the pathways in one of the clinics. There's also documented guidelines that have been published a couple of months ago as to when and who you should test and what types of tests you should be using. So it's really starting to come through into the clinical sphere. And in line with this, our team decided to develop a genetics clinic specifically for these patients so that we can facilitate appropriate testing because we can now see the benefit of this, and that in a significant portion of patients, we are actually finding a cause. So it's a multidisciplinary clinic model. We have clinical geneticists, genetic counselors, scientists, but also cardiologists involved. And I think it's really important for this patient group to have this involvement of all these different areas of expertise. So Annie Marish, one of my students, has did a quick review of our clinic, so looked how we're doing over a 2-year period. And what I really wanted to highlight here is the referral uptake is extremely high at 91%. So basically what that shows is that it really demonstrates the importance of this service that we've developed for patients and their families. What's also interesting to note is that the diagnostic yield is 33%, which is in line with other monogenic forms of disease. So congenital heart disease really should be regarded now, certain types of congenital heart disease should be regarded as a genetic condition for which we may be able to facilitate appropriate testing. Another study I thought I'd just quickly touch on is more clinical in focus. So Annie Morrish, again, she's doing an audit study looking at all the genetic testing that we've done over a 5-year period, really trying to understand which tests are most useful for our patients, which patient groups have the highest yield. And we're also including a survey to get an idea of what the value of this is to patients and their families, and if they have any preferences in how we continue to improve our clinic and the service that we're providing. And I won't go through this in a lot of detail, but essentially our preliminary results from this audit are sort of in line with what we're seeing in the literature, but also among our studies, that where we really are able to achieve a genetic diagnosis in over 30% of congenital heart disease patients with particular forms of disease. I also thought I'd quickly touch on the Kids Heart BioBank, which is a resource that has supported our genetic research now for about 15 plus years. And essentially it's a collection of DNA samples from patients undergoing surgery at the Children's Hospital at Westmead for congenital heart disease. And we collect their DNA and associated clinical information. And it really has facilitated everything that we have done so far, and has also been part of significant collaborations, both national and international, and really is the fuel to all the work that we do. And at present, we have about 4,800 DNA samples from patients and associated clinical information. So it's a really, really valuable, huge resource that we use. I also thought I'd quickly mention the study that Bridget O'Malley, one of my research assistants, is starting, and we're about to commence the study. But in line with what I mentioned earlier about thinking about the next generation, thinking about these kids as they grow up, it's really important for them to realize and have a better understanding that they are at a slightly increased risk of having a child with congenital heart disease, and being able to educate them in this regard. So what we're planning to do, or the ultimate goal, which I've highlighted here, is to develop a specialized education program for transitioning adults and adolescents with congenital heart disease. And in this survey, we're really just scoping out essentially a pilot study to scope out what their current understanding levels are, and also what preferences they have in terms of receiving this information, obviously with it being quite sensitive information. At the same time, we'll also be surveying clinicians via the Cardiac Society of Australia and New Zealand to understand their contribution in having discussions with adolescents and what their preferences might be. And lastly, I just thought I'd talk about some future work that I'm really working very hard at trying to get up and running, but essentially, again, looking at that next phase, looking at these kids as they grow up, and looking at maternal and prenatal contributors to disease development. So as I mentioned, a lot of the work that I've talked about today has focused on genes and genetic contributions, but there's this whole other side that we need to start looking into to really understand the full picture of disease development. And so the work that I'm hoping to do is a combination of sort of data linkage with some prospective studies, but really wanting to understand pregnancy and the factors that are actually contributing to the heart defect development, in addition to that increased genetic risk that they already have, in an attempt to hopefully modify the expression in pregnancies and hopefully reduce children born with congenital heart disease. And with that, I might end off. I'm sorry if I've talked a bit too long. I'm not sure what the timeline is, but I want to say thank you to my team, Kids Heart Research and the Heart Centre for Children, and the Victor Chang Cardiac Research Institute, who we collaborate with very heavily, specifically Sally Dunwoody and Eleni Yanalatu and their teams, and then obviously David Windlaw, who's now based in the US. Thank you very much.
Thank you, Gillian. So I'll just stop the screen share there, and we'll see if we can have any questions. But if not, I'll start first. So my question is, you're speaking about the dad who had a congenital heart defect, and then his son had even more severe congenital heart defect. What factors contribute to the expression of a gene being more severe or less severe? That's what we don't understand. So with congenital heart disease, we know there's that variable expression is extremely common. So we know that in one family, you can have someone with a very severe defect and they can have a child with a mild defect, or convert the other way around. And
we assume there's modifying factors, which could be genetic, but they could also be environmental. So it could be other factors that have affected the maternal, fetal, placental environment that are actually increasing or decreasing that severity. And I think the work that we're starting to do in that area is trying to discern what is the genetic, are there genetic modifiers and are there environmental modifiers, and how can we influence those to try and reduce the risk of severe disease.
So that's where your pregnancy work would come in.
Yes, pregnancy work will be looking very much more on the environmental side, and it will be on individuals who are at increased risk already.
I like the trajectory of your research and how it's,
plan and where it's going. There's a little bit of a pathway developing. Whether or not I get there, I don't know, but we're trying.
All right. Thanks, Gillian.